beginning of this whole meeting, and I'm Matt Hudson. So I'm a associate director Great Lakes with the Burke Center at Northland College, and so we do a lot of work with undergraduates who spend sometimes several years with us working on our various research projects, and Stephanie Wright is one of those people. And so Stephanie graduated last May, and she's been working with us full time since then. Many of you have worked with her through her work with beach monitoring and certainly with the cyanobacteria research that we've been doing in Lake Superior. So Stephanie is going to give this talk about our Burke Center work on looking at the risk of cyanobacterial blooms in the Chequamegon Bay region of Lake Superior. All right, the floor is yours, Steph.
Thank you, Matt. Well, as Matt mentioned, I'm Stephanie Wright, and I'm going to be discussing the research that the Burke Center has conducted regarding cyanobacteria in the Chequamegon Bay. So in previous nearshore group meetings, we have discussed our experimental work in 2019, and today I'm going to be updating you on the additional work that we've done in 2021. So we haven't seen any cyanobacterial blooms in Chequamegon Bay, despite the proximity of the bay to historical blooms and the relatively warm water temperatures that we have here in the summer. Next slide, please. So our research is based on the PhD work of Kate Reinl at University of Minnesota Duluth Large Lakes Observatory. So our 2019 experiments were designed to identify conditions and habitats favorable for cyanobacterial growth and possible blooms. We collected water, sediment, and plankton net tow in an effort to gather viable propagules. Our 2019 locations shown in the left map, we have 4 tributaries indicated by triangles, 3 coastal estuaries indicated by the squares, and 3 lake sites indicated by the circles. The culture samples were then incubated at 2 different temperatures with 2 different nutrient ratios in a triplicate for a total of 36 cultures. And then based on our results in 2019, we optimized the laboratory incubation conditions for 2021 and focused more on sampling locations. So you can see on the right map are lower tributary areas, estuaries, and shallow coastal habitats. Our goal was to identify specific locations around the bay that contained viable propagules for a bloom under optimal conditions. Next slide, please. So as you can see by the upward facing white triangles on the graph, our 2019 experiments showed cyanobacterial growth response from the combination of the estuary, the warmer temperature, and the low nitrogen to phosphorus nutrient ratio, rather than in our lake habitat cultures. So these results are similar to what Kate Reinl found in her research that we based our sampling on. So we were able to get a growth response, but the cultures with the response weren't visibly green. So the species dominant in the cultures with growth were Delicospermum, but they were different from the species present in the lake blooms, which were Delicospermum lemmermanii. Next slide, please. So that brings us to our very exciting 2021 results. So the majority of our locations under ideal laboratory conditions did not show measurable cyanobacterial growth, except for 2 estuary sites in the Bad River shown in the dark circles on the right hand of the map. So we weren't generating a dramatic growth response. So far, the evidence suggests that cyanobacteria are present in habitats around Chequamegon Bay, including the Delicospermum lemmermanii, but not in amounts where we have been able to make them bloom in what we have considered to be favorable laboratory conditions, and also in our sample location sites. Next slide, please. So we have microbial sequencing data from the 27 locations that we took in 2021, and it has just arrived, and it's currently being processed right now by Jake Callahan in Cody Sheik's lab at the Large Lakes Observatory. So hopefully this information is going to shed more light on which species are present from those 2021 samples. sample locations. They are also processing sequencing data from a series of nearshore samples collected from 31 different sites spanning from Superior to Bad River. In addition, we plan to collect sediment cores in 2022 to look for evidence of past blooms, and that's going to be with Katie Schreiner and Byron Steinman at the Large Lakes Observatory as well. So all of these data will hopefully shed more light on why blooms occur in some areas and not others. And then I just wanted to give a thank you to our funders at Wisconsin Coastal Management Program, and also to Kate and Bob Sterner and Sandy and everyone at the Large Lakes Observatory, as well as the Burke Center staff and student researchers.
Thank you, Stephanie. We're right on with 5 minutes. Way to go.